This topic is about microbiology of air. So, in this topic, we are going to learn about the common microbes that are present in air, their source and distribution, the techniques which are used for microbiological analysis of air, that means collection and then enumeration, counting of them, and then we will discuss about some measures or some techniques to control this um, microbial flora which is present in air. By the end of the topics, you will be able to describe the microbes that are present in air. You can discuss the significance of microbes that are present in air, and you can explain the process by which air sampling is done and how it is analyzed, and you will describe the process to control the presence of microbes in air. So, in if we talk about the air content, as you know, it is mainly made of gases, but in addition to those gases, dust particles, water vapor are also included in air content, and air also contains microorganisms. This may be the vegetative cells that are growing, or spores of bacteria, fungi, and algae, and it may also contains virus or virus particles or protozoan cyst. Since air is often exposed to sunlight, it is high in temperature, and it also contains less moisture as compared to soil and water habitat. So it does not provide that nutritive you can say supply for microorganisms so they can grow in that environment. So it's rather air is a transport medium for microorganisms. It is not uh, you can say very effective or very uh, favorable environment for microorganisms to live in. So that high temperature and less moisture tends to desiccate the living form, that is microbes, and most of them die. So it's only a transport medium for microorganisms as compared to soil and water. Now we can study the microflora of air under two headings. The first one is the outdoor microflora, the next is indoor microflora. The outdoor microflora means the air which is found outside the buildings, and the more dominant microflora of the outside air are fungi. Here we can see some examples. The sorry, this one, the fungi which are found in outdoor flora mainly consist of Cladosporium, Sporoblomyces, and the other genera which are included are Aspergillus, Alternaria, Phytophthora, and Eripsiphae. The bacterial species that are included in this outdoor flora contains Bacillus, Clostridium, Sarcina, Micrococcus, Corynebacterium, and Acromobacter. Outdoor flora also contains the uh, Basidiospores, ascospores of yeast, fragments of mycelium, conidia of molds. As air is present everywhere, and when air is blown, it may contain particles from the soil and water as a droplet in droplet form. So it carries those with its um, flowing. So that's why they are found in outdoor and as well indoor microflora as well. The number of microorganisms that are found in air, it may vary from place to place depending upon the human population and the density. Then comes indoor microflora. Indoor microflora is the air which is found inside the building or where a person is residing. The common genera of fungi which is found in indoor microflora contains Penicillium and Aspergillus, this one, and the commonest genera of bacteria found in indoor air is Staphylococci, Bacillus, and Clostridium. There may be variation according to the height or the place where a person is residing. So it is usually seen that the microbial content in the air becomes less with increase in altitude. Now I'll see how they are distributed in air. So as you know that no microbes are native to atmosphere, they represent allochthonous population. That means they are transient. They are not living inside that environment. They may come or they may go. So that's why with respect to aquatic and terrestrial habitat, they are not native. They are transient. They may come and they may go. So species vary greatly in their sensitivity to a given value of relative humidity, temperature, and radiation exposure. So more microbes are found in air over land masses than at sea. So microbes of air within 300 to 1000 or more feet of the Earth's surface are the organisms of soil that have become attached to the fragments of dried leaves, straw, or dust particles, and they are blown away by the wind. The spores of fungi, especially Cladosporium and Penicillium, Aspergillus, and Alternaria, they are more numerous, about 400 miles in both polar and tropical air, and at altitudes up to 10,000 feet. Many plant pathogens are also transported from one field to another through air, and they are the spread of many fungal diseases of plants. So the they can be predicted by means of the concentration of airborne fungal spores. We can predict the type of fungi that is causing the plant disease. Many human pathogens are also found in air, and they can cause illnesses such as tuberculosis, whooping cough, etc. They are also the cause of these disease. Then comes sources of microorganisms. If we are talking about where these microorganisms are coming and residing in air or found in air, there are different sources that contribute to the microbial air flora. The one common source is soil. The first one is soil. Soil microorganisms are distributed by uh, wind blow, and they remain suspended there for a long period of time. So, and activities such as digging or plugging the soil, they may also release soil-borne microorganisms in air. The next source is water. Water, the microorganisms found in water may also be released into air in the form of water droplets or aerosols. The splashing of water by wind action or the tidal action, they also produce droplets or aerosols. As you see that when uh, high tides are blown, and if you have experienced that tide, then small droplets that come on to your face, so they may also escape in the air, and they may contain those microbes which are found in water. The next one is the air currents. Air currents may also bring microorganisms from plant or animal surface um, into the air. These organisms may be commensals or plant or animal pathogens. So studies have shown that they can travel long distances. For example, spores of Puccinia graminis. It's a species that contains that can travel over 1,000 kilometers. However, the transmission of animal disease is not usually important in outside air. Then comes humans. Humans contain or they may also contribute to the microflora in air because they continuously inhale and exhale air. So that it might come in the form of droplet or droplet nuclei or infectious dust. The surface flora may be um, may be shed at times. The microbes that are present on your skin or that are on your clothes, etc. They may shed at times, and they may be distributed into air. Similarly, commensals as well as pathogenic flora of the upper respiratory tract and the mouth, they are constantly discharged into air by activities such as coughing, sneezing, talking, laughing. So the microorganisms are discharged out in three different forms, as I have included, mm, according to the size and moisture content. They may be you can say they may be classified as droplets, droplet nuclei, or infectious dust. Droplets are usually formed by sneezing, coughing, or talking. They each consist of saliva and mucus. It is estimated that a single sneeze may contain microorganisms between 10,000 to 100,000. So, they may contain hundreds of microorganisms which may be pathogenic if discharged from a diseased person. So, pathogens will be mostly of respiratory tract origin, and the size of the droplet determines the time period during which they can remain suspended in air. So, most droplets which are being exhaled out by a person, whether diseased or not, they are relatively large, and they tend to settle in still air, the air which is not moving. So, when inhaled, these droplets are trapped on the moist surfaces of the respiratory tract and become the cause of infectious disease. So, now uh, droplets are relatively large in size. So, what is droplet nuclei? Small droplets in a warm, dry atmosphere tend to evaporate rapidly and they become droplet nuclei. So, the residue of solid material which is left after drying up of a droplet is known as a droplet nuclei. ड्रॉपलेट न्यूक्लिया और ड्रॉपलेट में क्या डिफरेंस होगा? द डिफरेंस इज़ कि ड्रॉपलेट इज़ रिलेटिवली लार्ज, और ड्रॉपलेट न्यूक्लियाई जो है उसमें ये क्योंकि वार्म एनवायरनमेंट में होता है तो इसका जो लिक्विड पोर्शन होता है वो ज़्यादातर एवेपोरेट हो जाता है और पीछे जो सॉलिड रेजिड्यू रह जाता है दैट इज़ नोन एज ड्रॉपलेट न्यूक्लिया, एंड इट कैन सस्पेंड इन एयर फॉर अ longer period of time. They are small as compared to droplets, about 1 to 4 micrometer, and they are light. So they may remain suspended for hours or days, and they can travel along long distances as well. They become continuous source of infection if the organism is viable when dry. For suppose कि अगर कोई ऐसे स्पोर है या कोई ऐसे बैक्टीरियल या फंगल स्पोर है that can remain viable in dry form, so it become a continuous source of infection for persons who inhale those air. जो भी उस और environment में रहेगा या inhale करेगा तो उसके लिए ये एक potential source of infection बन जाएगा. Then comes infectious dust. These are large aerosol droplets that settle out rapidly from air onto various surfaces and get dry. Nasal or throat discharges from a patient can also contaminate surface and become dry. Disturbance of this dried material by activities such as bed making, handling of handkerchiefs, tissue papers, sweeping floors in the patient's room, they can generate dust particles which may add microorganisms to the circulating air. So most dust particles are laden with microorganisms. They are relatively large and they tend to settle rapidly. So microorganisms can survive for relatively longer periods in dust, and this creates a significant hazard, especially in hospital areas. So infectious dust can also be produced during laboratory practices like opening the containers of freeze-dried cultures, withdrawal of cotton plugs that have been wetted by culture uh, the culture fluids. Therefore, laboratory practices must be handled aseptically. That means they must be free from any contamination or any source of contamination from outside air. Infectious dust जो है ये आपके पास ये उन डस्ट पार्टिकल से भी प्रोड्यूस हो सकती है जो कि आप किसी एक ख़ास एनवायरनमेंट में, जैसे कोई नेज़ल ड्रॉप्स हैं या कोई ऐसी थ्रोट की कोई स्प्यूटम वगैरह है जो कि फ्लोर पे गिर रहा है और वो ड्राइड अप हो जा रहा है, देन इफ़ यू आर sweeping them, or you are cleaning those things, or you are carrying them from one place to another, तो वहाँ से भी microbes जो हैं वो air के अंदर escape हो सकते हैं, and they can become a potential source of infection. Several or the laboratory practices, maybe as you have seen in your lab as well, we put cotton plugs in test tubes. So if those cotton plugs are contaminated by the fluid, broth से अगर वो contaminate हो गया और उसको आपने निकाल के surface पे रख दिया, तो that surface will become contaminated as well. That's why it is always essential to follow aseptic practices in lab and in areas where there is a certain you can say potential of spreading microbes. So there you can also practice aseptic techniques. Then what are the methods by which you can analyze the microbial flora which is present in air, and how can you count them or enumerate them? So say there are several methods. Impingement in liquids, that means they are going to collect on the surface of a liquid, or impingement on solids, where they are going to collect or stick onto the surface of the solids. That contains sieve and slit method. So several methods which require special devices are designed for enumeration of microorganisms in air, and most important are solid and liquid impingement devices, filtration, etc. But none of them collects and counts all the microorganisms in the air sample tested. Some are destroyed and some are entirely passed through in all the process. So these are the different devices which are being used to collect and analyze the microbial flora that is present in air. First one is impingement in liquid. Impingement in liquid is done, the air is drawn through a very small opening or a capillary tube and bubbled through the liquid. The organism get trapped in the liquid medium. Then liquids of broth are plated to determine the microbial count. As you can see here, this is a cyclone sampler. The air is drawn from here, right? And then these are being circulated, and then there is a column which contains liquid. These the flora or the organisms which are being carried in this air will now drop or stick on the surface of the liquid. The second one is this swirling aerosol collector. Here you can see the air is being drawn in here, and then it circulates and then collects onto the surface of the liquid. Then you can take out the different concentrations of this liquid and plate them to count and incubate them, and then you can count the colonies, how many colonies are being produced by the given sample. Then impingement on solids, the microorganisms are collected directly onto the solid surface of the agar medium. And the most simple one is settling plate like this. You can uncover petri dish which contains agar, and then you can expose that petri dish in different areas where you want to collect the sample. Then after several hours or several minutes, you can take that petri dish, incubate it, and then you will determine colony count. Each colony usually represents the particle carrying a microorganism. It does not record the volume of air drawn, but it gives you information about the kind of microorganisms which may be present in a particular area. Then if you talk about the measured volume of air sampled through, then we have sieve and slit method. This is the sieve apparatus, and this is slit sampler. Sieve devices contains large holes, as you can see here. large number of small holes in a metal cover under which a petri dish is located, which contains an agar medium. So the measured volume of air is drawn from here through these small holes, and then air borne particles impinge or stick upon the agar surface. Then plates are incubated. Then plates are incubated and colonies are count. We have slit device also. The air is drawn through this slit. This is a slit sampler. Air is drawn through slit, a very narrow slit, onto a petri dish containing agar medium. Here this is the petri dish which contains agar medium. The slit is approximately the size of the petri dish, and petri dish is rotated at a particular speed under the slit, and one complete turn is made during sampling operation. यानी इसको फिर टर्न किया जाता है एगर प्लेट को, तो एक complete turn पे ये sample आपका withdraw हो गया, और उसके बाद आप इसको counting कर सकते हैं after incubation. We have then filtration also, जिसमें मिली पोर फिल्टर है. इट इज़ सेम एज व्हेन वी आर डूइंग वाटर एनालिसिस कि वैक्यूम पम्प के साथ आप जो है एयर को ड्रॉ कर लें, एंड देन यू कैन the microbes which are being present on the air will be impinge onto the filter. Then you can take out that filter and you can place them in a solid medium, and after incubation you can count the colonies. The filters which are used, they are biologically inert cellulose ethers. They are prepared as thin porous cellular membranes of about 150 micrometer in thickness, and the filters have different porosity and different grades, and you use them according to your need. So this funnel shape, it contains the assemblage where you can use these filters. They contain a funnel shape inlet and a tube like outlet, and in between these two the filter is fitted. The outlet may be connected to a filter so that it can suck a known amount of air, and then after collecting the required volume of air through that filter, it can be directly placed onto a solid medium, and after incubation you can count the colonies. Now why this air microflora is important, why their study is important, इनकी स्टडी क्यों आपके लिए इम्पोर्टेंट है, किन जगहों पे जो है ये आपके लिए ज़रूरी हो जाता है. एज़ यू नो दैट एयर इज़ इन कॉन्टैक्ट विद ऑल एनिमेटेड एंड एनिमेटेड ऑब्जेक्ट्स. हर चीज़ के दरमियान जो है एयर मौजूद है. सो एन एडल्ट मेल, इफ़ यू आर टॉकिंग अबाउट एन एडल्ट मेल inhales about 5 meter cube of air per day. And although most of the microorganisms present in this air is harmless, but less than 1% of airborne bacteria or pathogens as well. So although contamination may be low, but if the person is exposed to a high concentration of airborne pathogen, then he or she may become infected. And carriers which may carry these spores or these infectious dust within them, they may be symptomatic or asymptomatic, and they may continuously release respiratory pathogens in their exhaled air. The most common found pathogen in air is the Staph aureus. Generally, outdoor air doesn't contain disease-causing pathogen in significant numbers. However, purity of outdoor air is another aspect. The area in which you are living, the pollution, the pollutants which are being present in there, these all are another aspect of counting those microbial flora. In case of indoor air, the chance for the spread of infectious disease is more, especially in areas where people gather in large numbers, for example schools or theaters. So that is why microflora is a you can say is a source of a spread of infectious disease. And in this era of COVID-19, you can now understand why there is a restriction on mass gathering. ठीक है, इस स्कूल्स और थिएटर्स के लिए भी या किसी भी ऐसी जगह पे जहाँ पे बहुत सारे लोग एक साथ मौजूद हों, सो देयर exhaled air may contain those microorganisms and that it may become a source of infection for another person who is closely standing or close with that person. So this is why we are now um, focusing or now emphasizing on social distancing. Microflora in hospitals that may become it is also a very important aspect to learn. Hospital environment is usually thought to be free from microorganism, but it is not the case. Hospital environment can become a reservoir for pathogenic microorganisms which are being transmitted by patients, and such infections are called as nosocomial infections, which are acquired during hospitalization. So it can arise in a hospital unit or may be brought in by staff or patients admitted to the hospital. So, during hospitalization, people may become infected with other types of infections as well. So, this type of infections, which are acquired during hospitalization, are known as nosocomial infections, and it can arise in a hospital unit or it may be brought in by staff or patients which are admitted to the hospitals, whom are admitted to the hospital. Common microorganisms associated with hospital infections are Haemophilus influenza, Streptococcus pneumonia, Staphylococcus aureus, or Pseudomonas aeruginosa, which is the most common pathogen found in even it is resistant to certain kinds of disinfectant as well, and it can grow in there. So it becomes you can say continuous source of infection in other people. Some members of Enterobacteriaceae are also found in a hospital environment, and some viruses as well. So, development of high antibiotic resistance is also a potential problem among these nosocomial pathogens, as they are being contained in a hospital, and they are also exposed to different antibiotic uh, antibiotics as well, and therefore their mutation and their mutation can occur. So that is a very, you can say, source of antibiotic resistant in this environment. Then uh, we we have yes, microorganisms or microflora in industry. So as you know that air uh, is everywhere, and when it comes to industries, especially those industries which are manufacturing different fermented products, the if air contains microorganisms there, it may cause contamination of those products, such as alcoholic beverages or dairy products which are being made in different industries. They may also become a source of food spoilage, and as they are very troublesome in handling and in home and in industry as well. So food and other fermented products uh, which are prepared and packed, those areas must be sterile, and aseptic techniques should be followed in those areas. Now, if we look upon the physical technique which is used to control air microbe, there may be dust control, we can use UV radiations, we can use laminar airflow system. Dust particles contain microorganisms and allergens, therefore dust control is necessary in schools, home, factories, laboratories, hospitals, etc. So how you can control that dust? We can use a dry vacuum pump, which must be followed by application of suitable disinfectant or detergent solution. In addition, oiling of floors, corners, and uh, uh, oiling of floor corners also prove to be effective against dust control. UV radiation can also reduce indoor airborne microbial population. Germicidal lamps are used for this purpose that emits radiation in the range of 250 to 260 nanometer. They are usually incorporated in working bench, etc. We can also use laminar airflow system. This is an very widely used technique. This technique represents a unidirectional airflow system in which air passes through HEPA filters. HEPA filters are high efficiency particulate air, and it can remove particles as small as zero point 0.3 millimeters, zero point 0.3 mm. So it is very useful in industries where high degree of cleanliness is required for product reliability. It is also used in labs, and one such system is also installed in your lab, as you have seen it. And that's all from now. Now we have a quick recap. We can now easily distinguish between indoor and outdoor microflora, what types of organisms are present in indoor and outdoor microflora, how they are distributed in air in different heights, soil, water, air. What are the possible sources of air microflora, as we have seen, from soil, from water, from humans, and in humans, droplets, droplet nuclei, and then infectious dust. Then why this microflora is important? What are the significance of these microflora? As you have seen it, that they are significant, their study is significant because can become a potential source of infection and a constant source of infection as well. And as they can travel along very long distance, so that's why they are also causing uh, diseases from one field to another, as we have seen in plant pathogens. Then what are the techniques which are used to analyze microflora? We have liquid impingement techniques, we have solid impingement techniques, and then we have physical technique to control air microflora that contains dust control. We can use UV radiation, we can use laminar airflow system. For further reading, I would suggest you to go through the chapters which are given in Prescott's Microbiology and Pelixer. Thank you for now on. Now we'll do another session in another lecture. Thank you so much.